I just wanted to start out by giving you a little bit of history on the whole idea of events like this, because this is really an historic one, but it's not the first one of its kind. And it happens that the very first conference that dealt with shamanic plants, visionary plants, or what you might call hallucinogens, psychedelics, and so forth, and theogens, oddly enough, was organized by the United States government in an official capacity in 1967, at the beginning of 1967, 28 through 30 January 67, through the impetus of Nathan S. Klein at the National Institutes on Mental Health in the United States. And this conference was called Ethnopharmacologic Search for Psychoactive Drugs. And it had very much to do with the ethnopharmacologic search in the sense of the pharmaceutical company searching for new medicines, which of course I'll talk about in the context of Albert's work. And this brought together many of the leading experts on the subject. And this was 1967, and then Albert, of course, was a very young man of only 61 years old. And now he's aged somewhat since then. And sure enough, Sandoz wouldn't let him come to go to San Francisco. He was still an employee of Sandoz, and this was in the heyday of the bad publicity associated with LSD. And I guess Sandoz figured if they let him go to San Francisco in the summer of love, he might not come back. And so there was a big gap in this conference in that he was unable to attend. Then, of course, 10 years passed without any other further such conference. The United States government published the proceedings of this in a very good book, and it's one of the best values I've ever found in my library. It cost $4, non-copyrighted, but it's very difficult to get now. It's also called Ethnopharmacologic Search for Psychoactive Drugs. And therein there's a letter from Albert Hofmann expressing his regrets and a considerable desire to have been in San Francisco during this event. So 10 years went by, during which time Albert Hofmann retired from Sandoz, and a new generation of researchers that started more from the experiential side rather than an abstract way coming to the subject, myself included, had grown up and began to study this topic from this very unique perspective. And so the next conference was in 1977, 27 through 30 October, and that I organized with a couple of friends, and this was in Port Townsend, Washington. We actually had one the year before in 1976. But that strictly dealt with mushrooms, and it was a very small event and on a modest scale. But so we held this one, which was called the Second International Conference on Hallucinogenic Mushrooms, and it was right before that event that I was pleased to meet Albert Hofmann and his wife Anita for the first time. They stopped to see me. I was living in Olympia, Washington, south of where Port Townsend is, and they actually came on the Greyhound bus, which was really interesting to me because people in the United States usually wouldn't be caught dead on the Greyhound bus. It's considered to be really déclassé or whatever. But he came up from Corvallis, Oregon, so we met right before the conference. And there, similar to the event in San Francisco 10 years earlier, were several of his colleagues like Richard Evans Schultes, R. Gordon Wasson, and it also dealt with Sasha Shulgin as well, Weston Labar, Bo Homestead from Sweden, a great chemical pioneer from Europe also. And it also, Tim Plowman, Schulte's unfortunately late student, who was a specialist in coca. And it also dealt a little bit with the medical side of this subject, and Stanislav Grof was there, Norman Zinberg, Andrew Weil, and others. So that's kind of to set the stage, and there was another event the following year, 1978, which brought together many of the same researchers. And so luckily Albert Hofmann was able to attend those, and that was his first real connection with the US version of the psychedelic scene. And so that was a very interesting time for me especially. Okay, that's just a little bit of historical background. So this is now, that was then. And I have the somewhat difficult task of summarizing all of Albert Hofmann's contributions to chemical and pharmaceutical research, but I'm also going to talk a little bit about another aspect, which is, let's say, ethics, and also the role of intuition in research, because I feel he's made really significant contributions there by way of example. So I will start early on in his career and just work my way forward, and I'll try to leave time for questions. I don't want to make this too detailed in chemical arcana. Albert Hofmann showed signs of considerable promise very early on in that he got his undergraduate degree and his PhD in only a few years of study. And he entered the University of Zurich as a doctoral student and got his PhD with distinction in only one semester with only 3 months of research. And he achieved that by solving what was an important problem in natural products chemistry in his day that had been worked on by professionals, professors and so forth, unsuccessfully. And that was he solved the structure of chitin, which is the structural material of which the exoskeleton of crustaceans and insects are made. So by getting the stomach contents from a snail that lives in vineyards, he was able to degrade chitin and show that it was a polymer of glucose. And so that was his thesis. He was given this with distinction. And then he, of course, received many job offers, and he had 3 job offers in all. And he chose the one that paid the least. And this is another thing I will come back to, which is also significant, because he was following his heart. He wanted to work with plants. That had always been his dream. And the other 2 job offers that paid better and maybe were more convenient involved synthetic chemistry. And so he chose something because he wanted to do it, not because this was what was the most money. So he began working for Sandoz. He got his PhD in 1929 and also began working for Sandoz that year. The first 6 years, Sandoz, by the way, is now called Novartis, as I'm sure most everyone knows, but then it was the Sandoz Limited firm. So for the first 6 years, he worked pretty much exclusively on cardiac glucosides, which are compounds that you may ring familiar, digitalis or digitoxins. Compounds like those that are in digitalis that are used to stimulate the heart, very toxic compounds, dangerous also, and it's very important to have the pure compounds as opposed to a medicinal plant extract so that you can control the dose. But he was working with a European plant called Mediterranean squill, or Scilla maritima, and he published a series of papers on that. And then in the middle of the 1930s, on his own initiative, he asked to work on ergot alkaloids. Now, I'll just say a few words about ergot and describe briefly a little bit the chemical history. This is a parasitic fungus that grows on mostly on cultivated rye, the most famous one, but it grows on virtually any cereal, and there are many species and a couple of different genera. But the cultivated ergot, or ergot of rye, is Claviceps purpurea. And this had been a major product of Sandoz. It was involved early on in history, at least 1,000 years ago, we can be sure. There were episodes of mass poisoning that looked like epidemics and were even thought to be an epidemic, some type of communicable disease, until the 17th century when it was found to be a mass poisoning because many people lived only on bread, or mostly bread, and it was contaminating the rye that was their staple food. And this was realized only in the 1700s, late 1600s, that it was a fungus growing on the rye. But even so, this existed as a problem in the world up until the 20th century. In what was the Soviet Union, there were outbreaks into the 20th century. It may have occurred in antiquity, but we can't be sure. And so there were early attempts starting in about 1816, 1875, were the first chemical attempts to isolate active principles from this. But the first really significant event was in 1907. The Britons, Barger and Carr, isolated the first crystalline product from ergot that showed pharmacological activity, but unfortunately it was more toxic than anything else, and they called it ergotoxine. And 11 years later, in 1918, Arthur Stoll, who was Albert Hofmann's direct superior or boss in Sandoz, isolated ergotamine, which was the first really successful pharmaceutical product in pure form from ergot. And this was used, has been used in various realms of medicine, but I'll say more about that when we get on in the story. And so after 1918, Sandoz had a good business in ergotamine, but they stopped researching it at some point in the 1920s. And so Albert went to Stoll, his boss, and said, I want to work on ergot. And he said, fine. And so then he immediately requisitioned a half a gram of ergotamine from the production department. And his boss showed up in the lab very angry and said, you can't have a half a gram of my expensive product. You better find some other way to get your starting material for your research. And so he turned to the ergotoxin, which Sandoz also produced, and it was kind of like secondary waste matter in a way from the ergotamine production. And so he began studying this ergotoxin mixture at the same time as he was trying to isolate, break it down, and get pure lysergic acid. These alkaloids, well, the other significant chemical fact about them that precedes this is that very year, 1935, Jacobs and Craig in the United States had shown that lysergic acid was the building block of all of these alkaloids in ergot. And the name lysergic comes from lysis or cleavage of ergot alkaloids. And so they broke them down, and they had this common nucleus, lysergic acid, and I'll say more about that. And so alkaloids like LSD are built up from lysergic acid. And Albert was isolating lysergic acid for his chemical synthesis experiments from this ergotoxin. But in the process, he showed that ergotoxin, although it would crystallize, this sometimes happens in chemistry, was not pure and homogeneous. But in fact, it turned out to be 3 different alkaloids, really 4, because one of them had 2 isomeric forms. And so that was a very significant finding. And one of the alkaloids was already known and had been isolated just around 1935 also, and that was ergocristine, also in Sandoz. But the other 2 turned out to be new, and that was ergocorinine and ergocryptine. And ergocryptine was later found to exist in 2 mirror images, 2 isomeric forms, alpha and beta. So Albert was able to get this lysergic acid starting material while he was in the process of separating these 3 components or 4 components of the ergotoxin mixture. And so his first big project with this was in 1935, not only did Jacobs and Craig find this lysergic acid, but in 4 different laboratories, one of them being the Sandoz laboratories, but also simultaneously in the United States, in France, and in Britain, and here in Basel. Another alkaloid was isolated from ergot that is called mostly ergonovine. It has like 4 names. Here they called it ergobasine. But to eliminate the confusion, and because 4 people discovered it at once, they just put a new name, which is the new ergot alkaloid, ergonovine. And this is distinctive from ergotamine and the ergotoxin alkaloids in that it is water soluble. The others are soluble in organic solvents. And also that it showed the specifically uterotonic or oxytocic effects of ergot. And by that I mean one of the common uses and what led to scientific study of ergot is that it was actually in the United States that this entered academic medicine. John Stearns, a physician from Philadelphia, learned from a German midwife, Hofmann, that ergot was used to induce childbirth, to induce uterine contractions. And this effect is called uterotonic, or we now also call it oxytocic because there is a hormone, oxytocin, that produces this effect. So it acts like oxytocin on the uterus. And so, but this had tremendous value in obstetrics, and this was not toxic, not at all, not like ergotoxin and ergotamine with significant toxicity problems in higher doses. But the problem was it didn't occur, it was a trace alkaloid in ergot. And so, so Albert's first synthetic project was to make ergonovine synthetically. And he did this in 1936. And in the process, Jacobs and Craig had proposed its structure was lysergic acid propanol amide. And he proved this to be the structure by making it synthetically. And so this had significant economic value because now this ergonovine could be made synthetically. But unfortunately, it couldn't be patented because it was already in the open literature and there were many pretenders to the throne. So he went on synthesizing other alkaloids. Now, in LSD, My Problem Child, LSD, Mein Sorgenkind, Albert, I think, is a little excessively modest in that he takes credit for having developed 3 pharmaceutical preparations in his career, or what he calls successful pharmaceutical preparations in his career as a chemist. Well, I'm going to present the idea that I think there were really 9, not just 3, and I'll enumerate those and say a little bit about them. So, as I said, he made already in 1936 ergonovine, or ergobasine as they called it in Sandoz, synthetically. This was the first synthesis ever of any ergot alkaloid. So that's a significant thing in chemical research. It was also proving the structure of this alkaloid, because the way you prove the structure is you say, okay, I think it's made of these elements, so then you build it up from those elements and show that you can synthesize it. And it was of economic value, as I said, because now trace alkaloids could be produced from waste matter like the ergotoxine mixture. Okay, so then Albert went on. Meanwhile, he had separated out the 3 alkaloids from ergotoxine, the ergocornine, ergocryptine, and ergocristine. And so he made dihydroderivatives of those, and that's the famous mixture hydrogene. And this was a tremendously valuable product which could be patented and was patented, and which made many, many, many billions of euros, dollars, euros, whatever you want to denominate in for the company. And in fact, hydrogene has been off patent since already 40-some years, and it's still one of the top 20 leading pharmaceuticals in the world. And I'll say a little bit more about that business side later. So this was, this is a mixture of the dihydroderivatives of these 3 ergotoxine alkaloids. Now it still sells, as I say, in the top 20, but it must compete with many generic versions, copycat versions, because the patents have long since run out. And by analogy to this, about at the same time, he made dihydroergotamine, which is a variant, a semi-synthetic variant of the ergotamine alkaloid that Stoll discovered. That was also patented and is also still on the market as a pharmaceutical, although it's not as big a market as hydrogene. That's dihydroergotamine, and that's used as a circulatory remedy and for stabilizing blood pressure. And more or less in these same years, in the late 30s, and this is all pre-LSD days, he also synthesized a variant of this ergonovine, this water-soluble alkaloid, which came to be called methergine. And that's, instead of the propanolamide of lysergic acid, it's the butanolamide, so it has one more carbon atom tacked on the amine group. And this is the 3rd one that Albert takes credit for in his book, and this is indeed still on the market. And it's used in obstetrics, and it is mainly used now not for inducing uterine contractions, because oxytocin itself is used for that, but it's used to stop postpartum hemorrhage. And it's a very effective life-saving medicament for hemorrhage in the uterus after birth. So these are the 3 medicines that Albert took credit for, and indeed he developed them single-handedly, all by himself. I mean, and nowadays, if someone is lucky in a long career as a pharmaceutical chemist, they might work on a team that makes one drug that it gets approved, and there surely are some other examples, but it's almost singular that one single chemist working by himself would have produced 3 products that were patented and indeed are even still being sold many years after the patents have run out. So, but I said there were 9. These are only 3. Well, as a direct outgrowth of this research, even though Albert maybe didn't do the final steps of the synthesis, but for example, there was another product called Sansert or Deseril, which was made by his assistant, Troxler. And this follows along in the same range of research and working under Albert, although he was the first author on the paper. And this is the famous, it's been called anti-serotonin. And it's been at least as significant as a research tool as it has been as a medicine, but it's used as a medicine as an anti-migraine. So that's, there are three groups of three. So first we had hydrogene, methergine, and dihydrogot. So now we have the Sansert, or Deseril, as it's known in different countries under different names. Now, then there's another one, Parladel, or Pindolol. It's also, CB154, or Parladel. And this is going back to the ergotoxin. Sorry about the chemical nomenclature, but I said there was this ergocryptine was one of the three alkaloids you separated out from this mixture, and that had two isomers, alpha and beta. Well, this is a 2-bromo derivative of alpha ergocryptine. And it was called CB154, and it was made in this same series, and Albert Hofmann did personally make this one, but by an odd quirk of fate, this is an interesting point, they were lacking some data or had gotten lost or something when they published the paper in 1943 on all of these compounds. And once you publish it in the open literature, you cannot patent it. And so because they didn't have the data, they didn't publish it in 1943, and only much later did they find a use for it. And so had they published it in 1943, it might never have become a successful pharmaceutical because no one knew what to do with it in those days. But as scientific research advanced, much later, it was found to be a potentially very valuable compound, and it's used as an anti-Parkinsonian. And so it came to be patented under the name of another worker much later when Albert was around retirement age. And so, but that's also certainly to his credit, so that's the second of the second group of three. And similarly, the same co-worker, Troxler, but this time with Albert as co-author, made another successful patented drug called Viscan or Pindolol. And this happens to be a 4-hydroxy-indole derivative that came directly out of the research on the mushrooms. The psilocin is the active principle of the mushroom. That's 4-hydroxy-DMT. And 4-hydroxy-indoles are not at all common in natural products. This simple indoles, and this happened to grow out of that. And so, and this was also patented much later. And Albert mostly gives credit to his co-worker Troxler for this. But this is used as an antihypertensive and also in glaucoma. So then we have this second group of three, basically, successful money-making drugs. And I don't know if you know much about the pharmaceutical industry, but in most countries now it's very difficult to get a new medicine approved, and many people will work their whole career and not have anything but dry holes. But so that's the second group of three, Sansert, Parladel, and Viscan. Okay, and so there are three more. Well, how about Delicid and Indocybin? Do those ring a bell? Delicid is the trade name for lysergic acid diethylamide, D-lysergic acid diethylamide. And indeed, Sandoz intended, this was patented in the same paper right before they published that 1943 paper, and so it was on patent up until about 1960 or so. And Sandoz intended to sell this as a medicine, and it's a fabulous one. Take it from me, or you probably don't have to take it from me. But what they had in mind was two very good ideas. One of them was they had these 100 microgram, famous, Christian showed a slide, of ones that had already had the top taken off and were empty. But they made 100 microgram ampules. And they could be injected, and sometimes they were injected, but it was basically designed for oral ingestion, and it was just a protective measure to put it in the glass ampules against oxidation. But also they made, and the therapeutic indication for this in their prospectus, which they made and published in several languages, was twofold. It was for psychotherapy, for an aid in psychic loosening, as was mentioned earlier by Ralph, I think, this afternoon, and also possibly in the training of psychiatrists, because they had this idea of the model psychosis and so forth, and also maybe, give psychiatrists a taste of their own medicine. Let's make them, take it first. And so I had no offense to psychiatrists. And so that was the indication for the 100 microgram ampules. And the other one was 25 microgram pills. And this was designed as a stimulant for depression. This is brilliant, and this is something that we really need. Because at that dose, it's very much like amphetamine, but without the side effects, without any nervousness, a slight hint of something other than just the stimulation. But it's very difficult to tell the body effect of a low dose of LSD from a good amphetamine like methamphetamine. And yet it didn't have quite the tolerance problem. And so that was, Delicid certainly deserves to count. And also psilocybin. The same thing was repeated later. I mentioned in my earlier talk that the mushrooms that Gordon Wasson had rediscovered with Maria Sabina in Mexico in 1955 found their way here to Basel, and Albert isolated psilocybin and psilocin, and Sandoz, for similar reasons, intended to market psilocybin as indocybin, Indo for Indol and also for Indians. So they had 5 milligram pills of indocybin, and then they had 25 microgram pills, and also the 100 microgram solutions of Delicid. And so I don't think there's, even though Sandoz certainly didn't make any money off of indocybin and Delicid, I know a hell of a lot of other people that did make a hell of a lot of money off of it. And I don't think many people here would deny that these were really successful pharmaceutical discoveries by Albert Hofmann. And so he certainly gets, so that's 2 more, we're up to 8 now. And what's the 9th one? Well, oddly enough, reserpine. This is another interesting story about bad business judgment on the part of Sandoz. It happens that Albert started studying Rauwolfia serpentina also in the 50s, the snake root, and he historically, although he doesn't get credit for it because it's the publisher perish rule, but he became the first one historically to isolate reserpine from the Rauwolfia serpentina, the snake root. And the people at Sandoz said, no, that's no good. We'll just shelve that. And so he doesn't even get to publish it. And so meanwhile, Ciba patented it in 1958, and it was a revolutionary medicine in early psychiatric chemotherapy. So those are the 9 substances that, so really he participated directly and in some cases single-handedly, because indocybin and Delicid were also virtually single-handed, and reserpine, in the discovery of 9 successful major modern pharmaceuticals, basically all of which are still on the market in one form or another. Okay. Yeah, I can see I'm going to run out of time here. Well, that's not unusual. Okay, so I'll just say a couple more words about and other scientific discoveries along the way, and this is a little bit more technical, but I wouldn't want to leave it out. In the process of doing this synthetic work, people were puzzling over the structure of lysergic acid because it's not a simple compound at all. And especially the peptide alkaloids like ergotamine and so forth. And so Albert and his group basically derived the correct structural formula and the stereochemistry, which is to say the 3-dimensional configuration of something that is asymmetric, which means it can have mirror images that are not superimposable but have the same relation of parts for lysergic acid. And so that was a major discovery, and I know it's a point of pride to Albert that the American chemist Woodward, who won the Nobel Prize, was a competitor, and he had proposed a wrong structure, and which Albert was later happy to correct. And also, he later conducted with his group at Sandoz the total synthesis of ergotamine to prove this, this structure and stereochemistry. So those were major contributions in natural products chemistry that don't have this economic or business or medicinal dimension to them. And finally, on that note, he also participated on the chemical side in the development of the fermentation of ergot alkaloids, because in the early days, they started using wild ergot that came mostly from Spain and Portugal. And so this was separated out of the rye. Anytime you grow, I used to grow mushrooms on rye grain, and you can get, from seed rye, you can get a couple handfuls of ergots out of a 50-kilo sack. And so if it's produced for seed or for animal feed, they don't take the ergot out. If it's for bread or for whiskey, in Canada they just grow it for whiskey now. they take that out. Maybe they should leave it in. Who knows? And so, so this was done for many years, but then as the volume of sales grew with these alkaloids, thanks to, in no small measure, to Albert Hofmann, they began to intentionally inoculate rye fields with ascospores of the claviceps and hand collect, here in Switzerland, hand collect the ergots. So they contracted farmers to grow rye and intentionally infected. I'm sure the other rye growers in the neighborhood loved it. And intentionally infected the rye fields. But it's a big market now. It's produced in ton quantities. This was not small, though. Even when it was wild collected ergot, it was a couple hundred tons a year were collected and sold. And so not something you carry in your Volkswagen. And so, so they developed a procedure to grow this in big tanks in a factory, just as penicillium mold would be grown for producing antibiotics. And so Albert participated in this also, and there was intense competition. There were patents from several different companies, but Sandoz came out on top. And they ended up, through Albert's screening, finding a strain of ergot that produced almost pure lysergic acid with nothing else. It had a slight variant from lysergic acid and not a profile. Often there can be 20 alkaloids in these strains. It was from Portugal also, I believe. And so now it is produced in a factory in Germany in ton quantities. Okay, in closing, I want to say something about, I mentioned the subjects of intuition and ethics. I'll leave out the bioassay side because I don't have the time, and surely other people will talk about it. But Albert also advanced greatly the Hefter technique that Dave Nichols mentioned this afternoon. I call it the Hefter technique. Well, he mentioned in the context of Arthur Hefter doing self-experiments with the 4 alkaloids he had isolated from peyote, Lophophora williamsii, in order to ascertain which would be the visionary principle of the cactus, because you can't see this effect in research animals, and it's not so easy to see it in a friend even, from the outside, maybe even not in oneself. And so I suppose you can, I have once forgotten that I've taken it, but not for long. And so Albert did a whole series, at least 30 compounds he tested systematically, and unfortunately I've not been able to get him to write this up. I've even offered to do the heavy lifting because he's only written up the ones that worked, but we need to know the ones that didn't work also, save a lot of time and drilling dry holes in the future. Oh yeah, finally one more thing before I close with the intuition and ethics. He played a significant role in the Salvia Divinorum story also, in that he went to Mexico in 1962 with R. Gordon Wasson, and they brought back identifiable specimens. This had been known for a couple decades before this, not well known, but it was not identified botanically and named until they brought these specimens back, but they brought back live material that was cultivated, and all the subsequent chemical work done on it came from this live material, and this was sold on the plant market in the early days, even back in the 1970s. It was available, well before there were smart shops or shamanic plant dealers. And so that's another significant thing, even though he did chemical research on the salvia that was not fruitful. He did the bioassays there in Mexico, but the extracts he brought back with him following the lead of the Maztecs didn't work. And so he couldn't see the activity in his lab, so he was not able to isolate the active principle. So in closing, I just want to say, mention 2 things. One, as far as his contribution to the role of intuition in research, and the other as far as his contribution to ethics in research. As to intuition, the LSD story is striking. I mean, the textbooks of scientific history are going to have to be changed, because they commonly give as the classic scientific example of intuition in research Auguste Kekule's hypnagogic state on a streetcar or tram, and then he fell half asleep, and he had a dream of a serpent with its tail in his mouth, and this gave him the clue to the structure of benzene, because people were trying to figure out how the 6 carbon atoms of benzene were interrelated, and nobody considered that they might be in a ring, that these things could close on themselves and be cycles. But Albert's story is much more impressive than that. in that he had synthesized this molecule, and everyone celebrates the history based on when he discovered the effect, but in fact it was first made 5 years earlier in 1938. And so we've celebrated the history dates kind of 5 years late each time. We need to add a 5 onto the, but that upsets people's sense of order with a nice, 50, 60, and so forth. So, so he made this, and then he gave it to the Sandoz. He followed the rules. He gave it to the pharmaceutical department, and they said, nah, this is no good. So they tried it, and it was uterotonic, but the other compound they already had was better. It was stronger, so no advantage there. And so there was nothing interesting in these research animals to them. But the only research animal that they had that was capable of discovering this effect was Albert Hofmann. And the only reason he stepped up to the plate to do that is because of intuition. So he said, I've talked to him many times about this, and sometimes he just spontaneously brings it up. But he said things like, and these are more or less direct quotes, he liked the structure, or that he would have dreams about the structure, visualizing, the way he thought the molecule would link up and look and so forth. And so, so this is the example of intuition. And he said he had a striking presentiment that it had some activity they hadn't discovered. So he made it again in 1943, and that is when he accidentally, quote unquote, became inebriated and discovered the activity. And finally, I want to cite in conclusion for the ethical side, well, first, the ethics of choosing the job that pays the least, and where would that have led him? You can see where it led him here. On 11 October 1962, he conducted a very classical scientific experiment, which to my knowledge is unparalleled in the long history of pharmacognosy. And that is, he went back to Mexico with R. Gordon Wasson to seek out Maria Sabina, who had given Wasson the knowledge about the mushrooms that enabled Hofmann to isolate psilocybin or indocybin. And he brought back the pills of indocybin to Maria Sabina to test them on her, who was really a collaborator in this research. And so to him, the only way to really validate this isolation work was if Maria Sabina gave the okay to this substance. 11 October 1962, and she did. And so I'm sorry, I think I didn't leave time for questions, but thank you for your attention.